Hello everyone, and welcome back to the second part of the circulatory system lecture. So in the previous lecture, we've concluded our discussion discussing the mechanism of oxygen and carbon dioxide transport by the blood. And now here in the second part, we will go over the coagulation mechanisms, discuss the different nervous and endocrine control of the cardiovascular system, and then finally go over the lymphatic system, its functions, as well as its structure. So what happens when we have a blood vessel and there is a damage to this blood vessel? Blood will start flowing out of the vessel, and so therefore we need the clotting mechanisms in order to stop excess bleeding. So there are different mechanisms that will be initiated, and these mechanisms are usually being initiated by the endothelial cells. So we discussed how endothelial cells are not only essential for nutrient and gas exchange as well as formation of new vessels, they are also important for activation of the coagulation and clotting mechanisms. And so there are multiple mechanisms to stop bleeding. Number one is vasoconstriction. So once there is a vascular injury, there is a transient arteriolar vasoconstriction that is being induced by the release of endothelin from damaged endothelial cells, and consequently the local vasoconstriction of arterioles will slow down blood flow, and so therefore prevent excess bleeding. And then the other two mechanisms are primary hemostasis and secondary hemostasis. Before I go over those mechanisms, first I would like to stress on the difference between hemostasis and homeostasis. So basically homeostasis is maintenance of a stable equilibrium by physiological processes, versus here we are talking about hemostasis, which means arrest of the bleeding. So I thought it's important to go over those. In any case, so we discussed the first mechanism is via vasoconstriction. The second mechanism is by primary hemostasis, where the platelets will come and block the site of injury. But these platelets are very weak, and this blood clot can easily break off. So the second mechanism of strengthening a blood clot is via secondary hemostasis, where a fibrin mesh will strengthen this blood clot, and this is called secondary hemostasis. So let's go over these mechanisms. So we discussed vasoconstriction induced by release of endothelin. Then we have the primary hemostasis, where the platelets will bind to the subendothelial collagen that has been exposed after damage to the endothelial cells. And then there are von Willebrand factor or GP1B and other proteins that will help platelets bind to the subendothelial collagen, and then they would also help the platelets to bind to each other. So basically that's what I've shown you here, where the platelets will start to block the site of the injury. And then we need the formation of a fibrin polymer in order to strengthen this blood clot. And this is done via the secondary hemostasis. And secondary hemostasis can be activated by two different mechanisms, either the intrinsic or the extrinsic pathway. And so I will show you a memory aid how to memorize all these components. It's particularly important for your medical school to know the exact details. So once we have a damage outside the blood vessel, like for instance damage to the vessel wall or damage to any tissue, it will cause activation of the extrinsic pathway. So the name comes from it's outside the vessel, extrinsic pathway. So once there is a tissue damage, it releases the tissue factor that activates factor 7A, so it's activated 7A, that will activate the common pathways in order to initiate activation of thrombin. And so thrombin is the protein that will break down inactivated fibrinogen into its active form fibrin. So once fibrin is formed, these are monomers that with the help of the factor 13, these monomers can convert into polymers, and fibrin polymers will cross-link to form a mesh and strengthen the blood clot. So that's the time that a hard clot is formed. So one mechanism of activation of thrombin and fibrin was via the extrinsic pathway. The second mechanism is via the intrinsic pathway, where there is damage inside the blood vessels that releases kallikrein that causes activation of factor 12, factor 11, factor 9, and then with the help of the factor 8, these will activate factor 10, and then factor 10 with the help of factor 5 again will activate thrombin. Thrombin will activate fibrin, which will form a fibrin mesh. All right, now just going over all that, it may seem complicated, but it really isn't. So let me show you the memory aid that I use to memorize all this. So here I don't have any paper in front of me. It's just I just want to show you how once you understand the concept and use a few memory aids, it's so easy to recall the contents. So the numbers that I remember is that common pathway relies on factor 5 and factor 10. All right, and the other number that I memorize is number 7. And so I know that number 7 activates the extrinsic pathway. All right, so factor 7 activates the extrinsic pathway. And since the extrinsic pathway is initiated in the tissues, therefore it releases the tissue factor that acts on factor 7, and then factor 7 activates factor 5 and 10. And then factor 5 and 10, which are the common pathway, activate thrombin. And again, the number that I've memorized here, thrombin, is number 2. So factor 2A is being activated here. All right. Now, let's go over the intrinsic pathway. So we've already used up the numbers 10 and 7. So what other numbers are remaining? What's the number after 7? It's number 8. What's the number after number 8? It's number 9. What's the number after number 9? It's number 10, but we've already used it up for common pathway. So the number after 10 is 11, and the number after 11 is 12. So intrinsic pathway via the kallikrein, it activates factor 12, which activates factor 11, then 9, and then 8. And these will activate the common pathway that all of them again lead to the activation of thrombin. And then this is the important part. The function of thrombin is to convert the inactive fibrinogen into fibrin monomers. And then fibrin monomers, with the help of factor 13, will convert into fibrin polymers to form a fibrin mesh and strengthen the blood clot to form a hard blood clot. All right, so that's about it. That was a review of the clotting mechanisms. So again, we depend on vasoconstriction, primary hemostasis where the platelets bind together, and then secondary hemostasis via either the intrinsic or extrinsic pathway. All right, now once a blood clot is formed, at some point that the vessel is being repaired, we have to lyse the fibrin clot. We don't want it to be there all the time. And this is being done with the help of the plasmin. So that's an important protein that you should memorize. Plasmin is the protein that will break down the cross-linked fibrin polymer. It will degrade it into smaller molecules in order to dissolve the blood clot. All right, and this plasmin again is being activated with the help of the tissue plasmin activator or urokinase that converts the inactive plasminogen into its active form, plasmin. So once a blood clot is formed, some of the plasminogen is actually being trapped inside the fibrin fibers, as a consequence of which, once the tissue releases the plasmin activator or urokinase, this plasminogen will be converted into plasmin and break down the cross-linked fibrin polymers. So basically, in order to prevent excess bleeding or excess embolism, we don't want excess blood clot to be formed to block all the vessels. So therefore, the body needs to maintain a careful balance of the formation of blood clot as well as the fibrinolysis, which is destruction of the fibrin polymers. And so this is being done with the help of the plasmin for breakdown of the blood clot and with the help of the thrombin and fibrin for the formation of a blood clot. The next topic is the nervous and endocrine control of the cardiovascular system. So we've just discussed that the heartbeats originate in the sinoatrial node, and that sinoatrial node is being affected by the autonomic nervous system. So basically the sinoatrial node, in the absence of any extrinsic neuronal or endocrine control, has a rate of about 100 beats per minute. On the other hand, if this sympathetic nervous system acts on it, then this heart rate as well as contractility will increase. While if vagus nerve acts on it, then the heart rate and contractility decreases. And so therefore the sympathetic nervous system, since it increases our heart rate and increases contractility and blood pressure and so on, it's basically preparing us for stressful conditions, that we are ready to spend energy and take whatever it takes to come out of that situation. And so therefore this system is referred to as fight or flight response. And so the sympathetic nervous system is being activated with the release of the norepinephrine neurotransmitter that increases the heart rate and blood pressure. And so under these circumstances, we are ready to cope with this stressful condition somehow by either running away. So let's imagine that something starts chasing you, a dog starts chasing you, your heart rate is pumping, your blood pressure is high, you're ready to run. You feel like you've got energy going. And so that's the time that you can undergo flight response. Or at the same time, you're also ready to fight. So that's the whole purpose of the sympathetic nervous system, to prepare you for stressful condition. And so that's why the name fight or flight response is given to it. On the other hand, we have the parasympathetic nervous system, which becomes activated during the resting condition. So this is actually being activated during the resting condition, as a consequence of which it's sometimes referred to as rest and digest system. And so this parasympathetic nervous system, or PNS, is actually counteracting the actions of the sympathetic nervous system. Now let me ask you a question. Name a nerve that affects the heart rate via the parasympathetic nervous system. The answer is vagus. We already went over it. All right. And tell me, what neurotransmitters are being released by the parasympathetic nervous system? We already discussed that the sympathetic nervous system is releasing norepinephrine. All right. Now, how about the parasympathetic nervous system? What neurotransmitter are being used by the system? And the answer is acetylcholine is being used by the parasympathetic nervous system. So basically, upon the release of acetylcholine, there will be increase in peristalsis in order to enhance digestion. There will be increased secretion of the digestive enzymes, and then heart rate also decreases. Other findings that you could see is, for instance, contraction of bladder, to prepare you for urination, because that's the time to rest and digest. All right. Now, the next system that affects the cardiovascular system is the endocrine system. So in our endocrinology video, again, we went over all these topics, but it's always good to repeat to help you with the memorization. So let's go over all the hormones that affects the cardiovascular system, starting with adrenal medulla. Can you tell me what hormone is being released by adrenal medulla? So basically, norepinephrine and epinephrine. So you might have noticed that SNS uses norepinephrine as its neurotransmitter. Adrenal medulla also uses norepinephrine as a hormone, not as a neurotransmitter. Here it uses it as a hormone because it releases norepinephrine into the blood, and that's why it's called hormone. Adrenal medulla also uses epinephrine for regulation of blood pressure. And let me ask you another question. Where is adrenal medulla located? So if we have the kidney here, adrenal medulla is located right on top of the kidney. All right, so it releases norepinephrine and epinephrine into the blood, as a consequence of which it will activate the alpha adrenergic receptors and beta adrenergic receptors. And alpha adrenergic receptors cause vasoconstriction of the blood vessels, as a consequence of which blood pressure rises. Beta adrenergic receptors, on the other hand, increase heart rate and contractility. Next gland is the pituitary gland, and the posterior pituitary gland releases the hormone called vasopressin, the other name for which is antidiuretic hormone. And the function of this hormone is to increase renal reabsorption of water. It can also mediate vasoconstriction. So vasoconstriction and increase reabsorption of water in the collecting duct of the kidneys, as a consequence of which plasma volume increases, and so therefore blood pressure increases. Next we have the kidneys, and kidneys, with the help of the juxtaglomerular cells, they release renin in order to activate the angiotensin, and then angiotensin itself will activate aldosterone. So therefore here we have the activation of renin, angiotensin, aldosterone system, as a consequence of which there will be increase in blood volume and pressure. Next we have the heart, which releases the atrial natriuretic peptide by the atria and brain natriuretic peptide by the ventricles, and these are being released in response to the excessive stretching of the cardiac wall. So once there is high volume of blood, these will be released, as a consequence of which they would increase the glomerular filtration rate and decrease the SNS activation. And so increased glomerular filtration rate will decrease blood volume, and decreased SNS activation will decrease blood pressure. So this system is being activated in response to high blood pressure or high blood volume. And then finally we have the thyroid gland, which with the help of the follicular cells, it will release the thyroxine or T4 hormone, which increases the effects of epinephrine by increasing the number of beta-1 adrenergic receptors, as a consequence of which now there is enhanced cardiac pumping. So enhanced cardiac pumping or contractility. Next, I'd like to go over regulation of blood pressure. So in the previous video, we've already discussed how the blood volume is being regulated by different mechano or baroreceptors, and that's actually one very important mechanism of controlling blood pressure. So we discussed number one, by atrial mechanoreceptors, they would release the atrial natriuretic peptide in order to decrease blood pressure. Then we have the aortic and carotid mechanoreceptors that will activate the sympathetic nervous system in order to cause vasoconstriction or increase heart rate and to increase blood pressure. And then finally, we had the juxtaglomerular cells that will activate the renin, angiotensin, aldosterone system in order to increase the blood volume, as a consequence of which, again, blood pressure increases. So those were with the help of baroreceptors. Now, what we didn't discuss is that blood pressure can also be regulated with the help of chemoreceptors. So there are chemoreceptors that are located in the carotid and aortic bodies. And these chemoreceptors will monitor pH, so they would monitor pH, as well as O2 and CO2 content of the blood. So if there is decreased pH, meaning that there is high metabolic activity, or on the other hand, if there is low O2 because O2 is being used, or high CO2 from excess production of carbon dioxide, these receptors will become activated and inhibit the parasympathetic nervous system. So parasympathetic nervous system activity decreases while sympathetic nervous system activity increases, as a consequence of which there would be increased heart rate and increased contractility. And both of these mechanisms will increase blood pressure. Now, I would like to conclude the cardiovascular system by going over the blood flow in different vessels. And so, you know how if we have large diameter of a pipe and it becomes smaller, then here we have higher speed of the fluid here. So with the vessels that we are having here, the aorta, the vessel diameter is much larger than the capillaries. But then if we look at the blood velocity in the capillaries is lower. And the reason for that is that capillaries have a high cross-sectional area. So it's not just the diameter that is affecting the speed of fluid inside the vessels, but it's also increased cross-sectional of the capillaries, as a consequence of which the blood velocity is lowest in the capillaries. And here I've also included the equation for you for blood flow. Blood flow is being positively regulated by blood pressure, so the difference in blood pressure, as well as the radius of the vessel to the power 4. And then it's being slowed down by enhanced viscosity of the blood or increased length of the blood vessels. In this circumstance, however, cross-sectional area, when we are talking about the velocity of the blood, because of increased cross-sectional in the capillaries, the lowest blood velocity is in the capillaries. And it all makes sense. What's the physiological significance of having low blood velocity in the capillaries? So as you know, the function of blood capillaries is to allow nutrient, gas, and waste material to exchange between blood capillaries and tissues. And for that to happen, we need a slow blood velocity. If they rush through the capillaries so quick, nothing will happen. And so therefore, here by having low blood velocity in the capillaries, now it's possible for the gas, nutrients, as well as waste products to exchange between blood and the tissues. Alright, so that basically concludes our discussion of the cardiovascular system. The last section that I'd like to review here with you is the lymphatic system. And so basically with the lymphatic system, it has three main functions. So first, let's go over the components of the lymphatic system. Lymphatic system is composed of the lymph nodes, and lymphatic vessels. And number one function of the lymphatic system is to maintain fluid balance. So what happens in the blood vessels is that according to the Stirling's law, movement of fluid from blood vessel into the interstitial fluid is being influenced by two factors. One is the hydrostatic pressure in the blood vessels. So the higher the blood pressure, the more fluid is pushed out into the interstitial space. At the same time, blood contains many different proteins like albumin and other proteins that have some osmolarity, the other name for which is oncotic pressure, that wants to pull the fluid back inside the vessel. So the hydrostatic pressure is trying to push the fluid out, while the oncotic pressure is trying to pull the fluid inside the vessels. At the same time, the interstitial fluid itself also has a hydrostatic pressure that is trying to push the fluid into the vessels, and an oncotic pressure that is trying to pull the fluid inside. As a consequence of which, the net filtration pressure is actually hydrostatic pressure of the capillary minus hydrostatic pressure of the tissue or interstitial fluid. And then this value minus, now we are talking about oncotic pressure. Oncotic pressure of the capillary minus oncotic pressure of the tissue or interstitial fluid. So this equation will give you the net filtration pressure, and we are using the Stirling law for coming to this conclusion. So now the function of the lymphatic vessel is to pick up any fluid that leaks and remains inside the interstitial fluid. So imagine we didn't have the lymphatic vessel. What would have happened? The fluid that would leak out of our vessels will make our hands be edematous all the time. But that doesn't really happen unless you see someone that is really sick, like they have a heart problem that doesn't pump blood properly. Blood keeps staying in the vessels and leaks out, as a consequence of which their hands are always edematous. But in healthy individuals, you don't see that. And the reason is that that little fluid that leaks out, the lymphatic vessels will pick them up, and then they would empty this lymph fluid back into the cardiovascular circulation. So any fluid that is leaked out into the interstitial fluid or the tissues will be picked up by the lymphatic vessel. So the number one function was to maintain fluid balance. The second function of the lymphatic system is to absorb large macromolecules that cannot get absorbed into the intestinal capillary network. Like an example is fat. So inside our small intestine, we have intestinal lining with many villi and microvilli. And then the function of these villi is to absorb nutrients. However, these capillaries have basement membrane, and so large macromolecules or fats cannot get absorbed through the intestinal capillary network. The lymphatic vessels, however, don't have the basement membrane, as a consequence of which fat and other large molecules can get absorbed via the lymphatic vessels, the other name for which is lacteals. So I would like you to remember this name. Lacteal is the lymphatic vessels inside the intestinal network that allows for absorption of large macromolecules. And then the third function of the lymphatic system is to defend against foreign pathogens. So basically lymphatics are, I try to think of them as filters or police stations. Any fluid that enters the lymphatic system will have to pass through all these lymph nodes. And then the lymph nodes are major reservoirs of immune cells. They contain so many white blood cells, and so any fluid that passes through them will be checked by these systems, and so if there is a pathogen, they will be attacked by the immune cells. So therefore, the third function of the lymphatic system is to defend against foreign pathogens. Now, I'd like to go over different lymphoid organs. So first we have the primary lymphoid organs, which includes the thymus and the bone marrow. And these are called primary because the lymphocytes or white blood cells are being produced in these tissues but are not yet exposed to antigens. So bone marrow will produce B and T lymphocytes. B lymphocytes will mature in the bone marrow. T lymphocytes will go to the thymus and they mature there, but they are not yet exposed to any pathogens. So these are referred to as primary lymphoid organs. Then we have the secondary lymphoid organs, example of which include spleen, palatine tonsils, and lymph nodes. And it's inside these secondary lymphoid organs that T and B lymphocytes will get exposed to antigens and start fighting these bacteria or other pathogens. So let's go over every one of these, discuss what their function is. Spleen, we already discussed, has 3 different parts. Number 1 is red pulp that is engorged with red blood cells. Number 2 is white pulp that is engorged with white blood cells. So white pulp, and that's acting as a reservoir for white blood cells and then for fighting any bacteria. And number 3, splenic sinusoids for destroying old or damaged red blood cells by the macrophages. So spleen is a major reservoir for red and white blood cells, as well as a site for destruction of damaged red blood cells. Then we have the lymph nodes, and all the lymph that is being collected by the lymphatic vessels has to pass through the lymph nodes. And these lymph nodes are ideal for proliferation of white blood cells. So white blood cells that are inside the lymph nodes will get exposed to any pathogen that is being collected by the lymphatic vessels. And now from here on, the white blood cells can gain access to the cardiovascular circulation. And then finally, we have the tonsils, which are actually housing the lymphocytes. And once there is a viral or bacterial infection, like for instance in the kindergarten age, that's why the palatine tonsils will start to enlarge. And they may enlarge to a point that they may interfere with airflow into the upper airway, as a consequence of which you may hear that when I was a child I had some infection and they had to remove my palatine tonsils, and that's the reason for it. If it causes obstruction of the airway, then they would remove them. Okay, so we are basically done with the review of the cardiovascular system as well as the lymphatic system. So thank you all for watching, hope that helps, and we'll see you for the next video.